Hi, this is Miss Litton, and this is my wonderful lunch crew.
I'm using one hand to type, and one hand to get the pizza, so I'm not getting any other crew here.
And welcome to the review for today.
She complained about that earlier.
Okay. So today at lunch, glad you all got to hear that. We're going to review these items. Now, we have done reviews for each of those chapters already, right? Okay, so you want to go back and look at that. I'm just kind of pointing out this is what you need to make sure that you look over. And remember that the essays that I post here and on your worksheets are not, I'm not limited to those, so make sure you could get some from the back of each chapter. They have essays, right, that you could prep for. You could think up your own in order to respond to, but make sure you do that. So viruses, bacteria, and archaea, plant evolution and structure. That was a hyperdoc, right? These were, you have notes on all of it, but that was a hyperdoc. Plant nutrition and transport, was that a hyperdoc?
No.
No. Control of growth responses was a hyperdoc. And then flowering plants reproduction, we just discussed that. So I went and pulled from each of the prompts, you know, these are the essays that relate to that. So the first one says to diagram and describe the lytic and lysogenic cycles of a bacteriophage. That's a specific type of virus, okay? And what do they infect? Bacteria. When you do a diagram, okay, now on the AP exam, diagrams are only supplemental, but I oftentimes have you do diagrams because I can tell whether you understand it or not, and it's faster for me to grade. Truth be told, you need to make sure that your diagrams are good. Okay, I'm not asking you to be an artist, but if I can't see what's going on in your diagram, I cannot grade it. And so if you're going to do lytic and lysogenic, I might just ask for the lytic cycle, so make sure you can do that all by itself. I might just ask for the lysogenic cycle, make sure I can see that. Now the lytic cycle, I would make sure I have a stage for, what was the mnemonic we used for it? APBio. So what is A? Right, so if I was doing one like that, I would write number 1, attachment. I would draw a bacterium because it's infecting it, right? I would make sure I had a bacteriophage. Okay, I would label maybe that this is a protein capsid. Once I did that once, do I need to repeat that? No. Then I'm just doing this because I have, you know, something to deal with. I would label that that is a what? Yeah, a nucleic acid. Exactly right. Then I would probably have something in here that I would label is the what? The host chromosome. Right? And once I've labeled all of these things initially, then I don't have to redo it again, right? And so then I would do number 2. I'm not going to do all of it for you. What's number 2? What? Penetration. And so then I would repeat it. I would make sure I could see what was the difference between this one and the last one. And I would probably still have my here, and then I would have here, and I would show that this had come in here. You know, I'd have some drawing to show that the nucleic acid had come in there. And then what's after penetration? Biosynthesis. And I would see the different parts of the viruses that are made. The capsid, and I would say, look, these are a bunch of capsids. Here's a bunch of nucleic acids. Okay, then what comes after that? Maturation. I would see that there is a difference, okay, between biosynthesis and maturation. I would see that I could visually see that, and then release. Where could you find those pictures? In your what? In your notes, right? And about 1 million pictures online. Okay? And then if you were going to do the lysogenic cycle, you would need to know when it ditches out, or you know, when it comes back in. So make sure you review that. Any other questions on number 1 and expectations? Okay. Are you just sitting here waiting to log out because you're not staying for the review? Okay, bring it up to me and then I'll look at it while she's asking a question. Go right ahead.
When it attaches, does it actually release anything, or is it just attaching to that membrane and then penetration is the actual like injecting of the extra of their nucleic acid?
Yeah. So attach, it's just sitting there.
Okay.
Penetration, I would see it. I wouldn't have drawn that arrow like that. I mean, I would have just shown it in here.
Okay.
Yeah, and I would have like a little thing, like this is what happens during penetration. You know what I mean? I would have a little explanation of that. Okay, and try to see another hand up.
Can you go over the differences between biosynthesis and maturation?
Yeah, biosynthesis, they make the parts, and maturation, the parts are put together, but it's still inside the bacterium. So it's the parts, and then you put it together. It's like having the Legos and then putting the Lego together with the product.
So if you had a diagram, in maturation, it would be like all the little bacteria quadrants, but they're already assembled, they're still in the cell.
In maturation, yes. Yeah. Okay, any other questions on that? Okay, you would put that in the meeting before I opened it up, like you would put all the parts there, I wouldn't have it all over the place. Okay, number 2, explain the physiological mechanisms involved in conducting both water and nutrients through plant tissues in order that they can maintain homeostasis. In short, describe how both xylem and phloem function. Okay, so when you're talking about xylem, what would you say? It's called a what? What's the name? When you talk about xylem, you're moving water. What's that called? What is it called? Transpiration is part of it, but what do they call it when they talk about the movement of water? Maybe the TATC theory? Or the tension cohesion theory? I would be able to name it, okay, which nobody could in the room, okay? So I would name it, okay? And then what I would do is make sure if I tell you TATC theory, right, you would be able to talk about all 4 aspects of it, right? But before you start talking about it, you might want to set it up so I know a little bit about xylem, right? So I want to say things like xylem, it actually functions when it's dead. Yeah, it'll function when dead. In fact, it's best when it's dead, right? Of course I can. What is it? What am I eating?
Like Indian chocolate chip cookies.
Oh, okay, wait, I gotta check this out. I'm eating Indian chocolate chip cookies. I like Indian chocolate chip cookies. They don't have any fat in them, right? Okay, that's enough. I don't Okay, what group are you? 4? Gotcha. All right. So talk about xylem, talk about how it can function when it's dead and why it can function when it's dead. And then you break out down each part and the role that it plays. Okay? Yes?
Why can't it function instead?
Anybody want to answer that? I'm not going to write your essay for you. Yeah.
Because when it's alive, the tissues can get in the way of the water transporting, so instead it's not alive.
Yeah, you don't need it to be alive because there's no active transport of anything, right? It's basically just a tube. So then it's nice if the water is just bound to other water. What's that called? Cohesion or there. You know what I was saying? Yeah.
Could you explain the tension part of
Water always goes. It looks good. Eat it. Water always goes from a higher water potential to. Is it sticking to it? I don't think so. From a higher water potential to a lower, right? And so the highest water potential, if you're going to get water moving, the highest water potential is probably in the soil. And then it moves to a lower water potential in the root. Now, how did that lower water potential get there? Proton pump, then you could do an exchange, right? And so you make the steel the center of the root, very high protonic, so water wants to flow in, right? Okay. And then, remember, no active transport of water, right? Then there's a little bit lower water potential in the stem and a little bit lower, right, all the way up into the leaves. And then the lowest water potential of all is going to be where? Outside in the dry air, right? So water, the tension is due to the pull of transpiration on that water column. It's like a straw and you're sucking up the straw. That's the tension that you have that's continuous. As long as the water column is continuous, that tension will be continuous, right? It's like if we had a line between us and we could pull on it, there's tension, right? If the line gets cut and the hydrogen bonds aren't there between the water molecules, then you don't have that tension, right? So you want it to be like one continuous water column all the way up to the top and out. So that's based on water potential. Tension is about water pressure. All right, we okay with that part? Okay. Then what's the name? What does sugar flow through? Good. So what could you tell me about phloem structure? Because you'd want to set that up first. Sieve tube members and companion cells. What's the role of the companion cell, do they think?
The nucleus and the proteins. Like the proteins.
Okay, yeah, because it's assumed that the companion cell is controlling the sieve tube element because the sieve tube member does not have a nucleus, okay? But it's the thing that's moving the sugars, not the companion cell, right? The sugars are moving through there. So once again, how is that getting accomplished? Well, probably due to a proton pump, right? Creating a +1 environment where you can escort in the sugar. And you're concentrating the sugars from the source to the sink. And that's where you have an opportunity to tell me what is a source and what is a sink. So a source is where all your sugars are, right? And that could be due to the process of, or it could be due to the storage in the roots, yeah. So, for instance, the tree outside our room now is no longer green, so the source is going to be in the roots, right? And the sink is going to be the rest of the plant that the roots have all the sugar to keep the rest of the plant alive. That was reversed though, right? When we came to school, the leaves were there and they were the source, and the other one was the roots were the sink. Yes?
So the leaves produce sugars through respiration?
No. Photosynthesis. You know. Yeah. Okay? So source is wherever you're getting. Now, in the leaves are doing photosynthesis and they're generating those sugars. When it's in the roots, it's probably stored, you know, as a starch or some other compound, right? And it's going to be converted into glucose. So that process has to happen. So basically what you get is you get a bunch of sugars to move into the sieve tube element due to your proton pump. Then it makes that particular sieve tube element is what? High tonic. So then water wants to flow into the sieve tube element, right? And that increases the turgor pressure. So now that it's going to want to move, right? Turgor pressure, it's going to want to move. Remember when we talked about plasmodesmata, the cellular junctions in plant cells, right? So the wall is not completely continuous, but there's junctions between plant cells. And so it can move, the sugars can move from one area to another. It's not going to come from the source because the pressure is still high from the source, right? Because it has the most sugars. And just like, it's not tension, but do you remember how we talked about tension? Now it's continuous. It's instead of being a pull like you would with transpiration, it's a push. So it's always coming from one direction, the source. And it's like sugars are coming and water's following it, and sugars are coming. As soon as these sugars move from where there's less sugar, the sugar water moves from where there's more turgor pressure to where it's less, this gets filled up right behind it with more sugar put in there, more sugar put in there. And so it just keeps like, okay, all right, okay. And it just keeps getting shoved to the sink, the part where the sugars are getting used. Does that make sense? Okay. Any questions on 2? Let 2 go? Okay, number 3. Concerning plant reproduction and homeostasis. Diagram and label the alternation of generations as well as the parts of a complete flower. That's probably an old school that you're going to just the basics of the flower. Don't sit there and memorize all this. Remember how the only ones I said to talk about and like the reproductive organs, that's it, okay? You don't have to get every single part of a flower. I don't want you to memorize that. Should you know alternation of generations? Yes, okay? And you should know what's haploid, what's diploid, what structures make what. And that is not I don't understand, I don't think. It's more of a matter of I've either learned it or not. But it doesn't have to be about memorizing. You can just make it make sense. If I said to you, here is this plant, okay? Here's the big part of the plant, okay? What would you refer to this whole plant as? Yeah, I would call this the sporophyte, and is it diploid or haploid? So I would put 2N, Okay? And then what does the sporophyte always make? Spores. By what process? Meiosis, right? So meiosis is going to make spores that are what? Haploid. Okay? What did the spores grow up to be? Gametophyte. By what process does it do that? Good. Okay? That gametophyte, if I'm talking about a flowering plant, you know what that gametophyte looks like, right? The female gametophyte looks like this. I didn't draw it very good. Do you know what I just did there? And then the female gamete is actually right there, right? That is the actual gamete. Gamete. Right? This whole thing is the female gametophyte. Okay? And then the male gametophyte, okay, that would be like a pollen grain. And it has 3 sperm in there, right? What's the job of one of those? 3 nuclei. What's the job of one of them? Okay, one will fertilize the gamete. What about another one? Yeah, one will make the tube. What about the other one? Yeah, it'll fertilize, in a sense, that polar nuclei, making the triploid endosperm, right? So these are the actual gametes, okay, are right here and right here. But this whole thing is the gametophyte. This whole thing is your gametophyte. Yes?
So when we eat fruits, are we eating the female gametophytes?
What are we eating? The ovary that surrounds it. So if you look, I would probably draw, do you know what I'm drawing right now? Sorry. Do you know what that is? Okay, it looks terrible. But this would be the pistil, right? This would be the stigma. This would be the style, and this would be the ovary. Okay, inside of there is where you have your ovum, right? And you have your carpels, and then you have your mother megaspore, and then your megaspore, remember 3 die off, and then you have just 1 spore that actually develops, okay? So that's kind of messy, but then the gametophyte makes these gametes. The gametes get together through what process? Fertilization, and then they're going to form a, which is what? Which is going to grow up to be the sporophyte by what process? Mitosis. Okay, I am not expecting you, if I'm doing alternation of generations, to do the mother megaspore and then meiosis, and then you show me, you know, that only one of the megaspores develops into the female. That's a lot to put on a diagram. I'm not looking for that. I'm just making sure you understand that all of that is happening inside here. Yes?
So the only 2 diploid structures on there is the sporophyte, and the zygote.
Yep, everything else is haploid. Exactly. Okay. What else? Okay, so it says diagram the alternation generation as well as the parts of a complete flower. Identify, explain ways that plants maintain homeostasis through feedback mechanisms, think hormones. Okay, now, you don't have to know all the hormones anymore. What's the one hormone you need to know? Auxin. But you do need to understand about photoperiodism, right? And so that kind of lends itself better to these questions here. Okay, so, and these are the more modern ones that I gave you out of the Google Doc, right? Those were the PDF ones. So, things like photoperiodism and phototropism. Okay, photoperiodism is responding to light and dark, right? But phototropism is growing towards the light. So you want to make sure you understand the difference between each of those. And then it says, are plant behaviors that increase fitness? What does that mean when a plant's fitness is increased? Yeah, chance to produce offspring.
So there was this question, I don't know if it's on the PDF, it might have been part of like the written essays, but it said starting from a seed, describe the development of a unipotent embryo.
This one, somewhere in here?
No, it was the one who was in
Oh, okay, we haven't done seed germination. Yeah, that's a different chapter. All right, good, so good? Okay, so then it says, explain how chrysanthemums, which are short-day plants, can be forced to flower in a greenhouse in the middle of the summer, including your discussion, the specific signal, and its reception. Okay, so, interesting, explain how chrysanthemums, which are short-day plants, when do short day plants exist? During the winter when the days are short and they must need what? Long nights. But here's the part. Can be forced to flower in the middle of the summer when the nights are long. Are short. Thank you. In the summer when the nights are short and the days are long. Sorry. So how could that possibly happen? Any ideas? Make it dark what? Longer, right? You're controlling the environment in which they're flowering in. So you would, in order to talk about that, you need to say, well, normally when you have a short day plant, it's going to need a long night, okay? In the summer you don't have long, dang it, you have short nights. No, I was doing it right, and so you would have to create an artificial environment, so you would, that way you're kind of explaining, I can tell you understand the whole process. Okay. B, a plant seeding will respond to strong directional light source. Explain how this occurs. Okay, so then this brings us back to what I said. The only hormone you really need to know is what? Auxin. And what did we say about how that happens is that auxin will move to the shady side. Right. And when it's on the shady side, it's forcing those cells to do what? Elongate. Cytokinin makes you make more cells, but auxin makes them elongate. Okay, how does it do that? Proton pump. Once again, another purpose, right? And those hydrogen ions out in the cellulose make the wall weak, so that when water moves into the vacuoles, because normally the wall pushes back, that's called what? But now the wall is, it can't pull back, so it starts to stretch, and then you rebuild more wall parts and it gets longer. On the shady side, the sunny side stays the same length, and that forces it to turn towards the light. Okay, and that is called, you can look it up, acid growth hypothesis. Acid growth hypothesis, which makes sense, right? Acid, low pH, proton pump. Yeah. Okay. Questions on B? Yes.
I'll put the red light and the far red light for the.
Yes, please do. I thought the next question did. I'm reading it right now. And it doesn't. Yes, please talk about. So when we talked about, what were the two, our little toggle switch of our phytochrome was what? P, R, right? And P, and PFR, okay? And then during the daylight, that's when it gets converted back to PFR. And then during the nighttime, right, it goes back to PR. The longer your night is, do you see how you get more of it to revert back to the PR, the longer your night, because you have more time. In the summer, you don't have as much time. But remember, it's weird because, because it's not consistent, right? Certain plants need a certain amount of PFR if you're a short day or you're a long day. It's not consistent across all plants what they require for flowering. Some need a lot, some need very little. Okay. Yeah, talk about that here. Okay, talk about that in A if I ask that one. I would probably ask you like specifically on that one just to make sure you did, but yes, that would be good. Okay, explain how each response increases biological fitness. So anything that you you make those links together. Yes.
I had a question yesterday. Is bonus and autism and adaptation because it adapts to the plants?
Yeah, but the light is not only important for flowering. What else is light important for? Exactly. So you're maximizing the amount of time that your leaves are towards the sun, right? So you take advantage of it. So not just for flowering and reproduction, but it makes you more fit too, right? To be able to do more photosynthesis. Okay, so the last one, you could do that, right? Because we've talked about that has to do with phototropism and just changing two different environments. Okay, good job. I'm so glad we did this today. You're super smart. If you haven't signed me off on the document or on your lab, make sure I do. Have a piece of toast.